there, this is Dr. Rosario, and this is our second lecture out of Chapter 9. So in our last lecture, we discussed redox reactions and how they can be used by the cell to produce ATP, that energy currency of the cell. In this lecture, we're going to get into the details of those reactions. So we are going to identify the stages of cellular respiration, and then I'm going to focus in on the first half of cellular respiration, which is the oxidation of glucose. So we're going to, like I said, discuss the major stages. Why do we perform cellular respiration? Where cellular respiration occurs? What are the major reactants and products? And then how much ATP are we making in this process? I know I mentioned it in the last lecture and I will say it again. I am not concerned with you knowing each of the steps of these chemical pathways. I have a couple of figures where I give you an overview of the stage, and there you will see that I have identified the inputs and the outputs for each one of these stages. That's where I really want you to focus your attention. Don't get caught up in the details and the complicated chemical names and the crazy enzyme names. Focus on the big picture. What are we doing here? In cellular respiration, remember, we are taking the chemical potential energy that is stored in glucose and we're releasing it from that molecule and making ATP. ATP, again, is that energy currency of the cell. So, cellular respiration has two components to it. There is the complete oxidation of glucose to carbon dioxide, and remember we had this formula that I had put up on the screen during the last lecture. The second half of that formula is the reduction of oxygen. So there's two parts. Whenever you oxidize something, you have to reduce something else. In aerobic respiration, what we call the final electron acceptor is oxygen. It is the thing that is going to receive the electrons ultimately from the glucose molecule. Now, there are some organisms that can perform anaerobic respiration where they use other molecules besides oxygen to respire. We're not going to worry about that in this class. You can learn about that in a microbiology class, for example, because that's very common in our bacteria. But for this class, when you hear the word respiration, it is essentially equivalent to aerobic respiration. So there are four main stages for cellular respiration for this process of breaking down glucose, releasing the energy, and making ATP. Those four stages are glycolysis, which occurs in the cytosol, and then pyruvate oxidation, the citric acid cycle or Krebs cycle, and oxidative phosphorylation. Those three steps occur within the mitochondria of our eukaryotic cells. In all of these steps, we are removing electrons from glucose and we're going to transfer them ultimately to that oxygen molecule. We're also going to generate ATP as we walk through this process, but most of the ATP is going to be produced during that last stage, that oxidative phosphorylation. In this lecture, I'm going to focus my attention on these first three stages because this is where we see the oxidation steps occurring. In the next lecture, we'll focus on the reduction of oxygen, which is our oxidative phosphorylation step. Okay, so glycolysis, that word literally means to split sugar. So I'm going to draw my glucose molecules on the screen here for you, and I'm really going to only draw in the carbon atoms. The hydrogen and the oxygen, they are important, but I'm going to trace the flow of the carbon through this process. So in a molecule of glucose, you have 6 carbon atoms that are linked together through covalent bonds. This is an organic molecule. It has chemical potential energy. Through glycolysis, we're going to release a little bit of that energy, and we're going to break that molecule in half. So we're going to go from one 6-carbon molecule to 2 3-carbon molecules. The main product of glycolysis is pyruvate or pyruvic acid. Those two terms are used interchangeably. All right, so that's where the carbon goes. We will also remove electrons from that glucose, so we are going to do some oxidizing here, and those electrons are going to be temporarily placed on our electron carrier molecules, our NAD, right? NAD will pick up electrons and hydrogen to become NADH. There will also be some hydrogen ions that will be generated in this process. We will also get a little bit of ATP through glycolysis. So for each glucose that is fed into this pathway, we get 2 ATP, 2 NADH, and 2 pyruvate. The production of ATP here is through substrate level phosphorylation. Remember, that is the direct transfer of a phosphate group from an organic molecule to ADP to make ATP. Okay, so let's take a little bit of a closer look at what goes on in glycolysis. There are 2 stages, all right? It can be broken up into 2 halves. In the first half, we call it the energy investment stage. It is called this because we actually use a little bit of ATP in order to get that glucose molecule ready so that we can harvest the energy from it. And then the second half of glycolysis is that energy payoff stage where we actually are going to pull the energy out of the molecule. So, like I said, there's a lot of big chemical names here. There's a lot of big enzyme names here. I don't expect you to memorize those. What I want you to see here is what goes into this process and then what comes out of this process. So in the energy investment stage, we start off with that molecule of glucose. And the first step of this process, the first reaction, is one in which we take an ATP molecule and we remove the phosphate from that ATP, so we generate ADP, and that phosphate actually gets transferred to the glucose. So that second molecule you see here is glucose 6-phosphate, and you can see that the phosphate group is attached to the 6th carbon on that sugar. In the next step, it's just a rearrangement. And then in the third step, we take a second ATP molecule and we add it to the other end of the molecule. So now we have fructose 1,6-bisphosphate. Now, that molecule has a phosphate group at either end. That makes it possible for the next step to occur, which is that the molecule actually gets broken in half into 2 3-carbon molecules. I do want to draw your attention to the name of that molecule at the end called glyceraldehyde 3-phosphate, or G3P for short. That particular molecule is important because we'll see it in other chemical pathways later on in this unit. But the overall inputs and outputs for this first stage of glycolysis, glucose goes in, 2 ATP go in, and what comes out is 2 ADP and 2 glyceraldehyde 3-phosphate. In this process, the 2 ATPs are being used to activate our glucose molecule. Now, in the second stage of glycolysis, we're going to pull those phosphates back off of the molecule. And in doing so, we're going to regenerate that ATP that we used up earlier. So in the energy payoff phase, you'll notice here the first step of the energy payoff phase is an oxidation step. We pull off those electrons and transfer them to NADH. And then as we go through steps 7, 8, 9, and 10, we regenerate that ATP. We actually make 4 ATP per glucose in this stage. The final product is pyruvate. We get 2 pyruvate molecules for every glucose. Remember, glucose has 6 carbons, pyruvate has 3. So the electrons are transferred to 2 NAD to make NADH, and we make 4 ATP. Again, this is going to be through substrate level phosphorylation. So we can look at the net inputs and outputs of glycolysis. What goes in to begin with is glucose. In that first stage, we use up 2 ATP, but in the second stage, we regenerate 4 ATP, so our net gain of ATP from glycolysis is 2. We also get 2 NADH and 2 pyruvate. All right, now we're going to focus in on what happens to the pyruvate next. So glycolysis, here's the overall process. We're splitting our sugar, right? And we're getting those 2 pyruvate. So the pyruvate is then going to be oxidized further. This pyruvate oxidation step is also sometimes called the intermediate step. This is going to happen in the mitochondria of the cell. So we start with our 2 pyruvate that came from our glucose molecule, and then we're going to remove electrons from those pyruvate molecules, and in doing so, we produce 2 more NADH. We also remove 2 of the carbons from the molecule. They get released into carbon dioxide, which just evolves away from the cell. That is waste. That's what we breathe out. Coenzyme A is a carbon carrier molecule. So in this reaction, what is going to happen is the remaining 2 carbons from each of these pyruvates is going to be attached to coenzyme A. And that gives us acetyl-CoA. A 2-carbon group is called an acetyl group, and we have 2 of those acetyl groups attached to 2 coenzyme A's. Again, we're going to follow the flow of the carbon from glucose through glycolysis, through pyruvate oxidation. We now end up with our 2-acetyl-CoA. The next step in the process is what we call the Krebs cycle. So we're going to follow that acetyl-CoA as it enters into this process. So the Krebs cycle is also called the citric acid cycle. And here we're going to see the oxidation of that acetyl-CoA. So we start with those 2 molecules of acetyl-CoA, and immediately those carbons that were attached to coenzyme A are going to be fed into this chemical pathway, and the coenzyme A is going to be released. This is an oxidation, so we're going to see lots of electron carriers involved in the Krebs cycle. You have 6 NAD being reduced to form NADH, and you have 2 FAD picking up electrons and becoming FADH2. As we oxidize the acetyl groups, we release the carbon in the form of carbon dioxide. We do generate a little bit of ATP through the Krebs cycle, and this is again through substrate level phosphorylation. This entire process occurs within the matrix of the mitochondria, so that inner jelly center of that mitochondria organelle. So this is a little bit of a closer look at the Krebs cycle. The Krebs cycle is actually called a cycle because it begins and ends with the same molecule. That molecule is called oxaloacetate. What we're really interested in is the products of the Krebs cycle. So the products are going to be our 6-NADH and 6H+, 2 FADH2, 4 carbon dioxides, and finally 2 ATP through substrate level phosphorylation. Now, when you look at this figure, you'll notice down at the bottom, we're actually producing GTP, and which is then can be used to make ATP. So it's a little bit more indirect here. But again, this is going to be substrate level phosphorylation. You do not need to memorize the steps of the citric acid cycle. You do not need to memorize the individual enzymes that are involved. I do want to point out that the name of this cycle, the citric acid cycle, comes from that first molecule that is made, which is citrate. Okay, so we have discussed now the complete oxidation of glucose to carbon dioxide. We took that glucose molecule and we converted it to 6 carbon dioxide molecules. In the process, we picked up electrons on NADH and FADH2, and we produced a little bit of ATP through substrate level phosphorylation. Now, where is the energy that was in that glucose molecule? Some is found in the ATP, but most of it is found in those electrons that are on those electron carriers. And in our next lecture, we're going to discuss how that energy then gets converted to ATP through oxidative phosphorylation.